Today, we are talking about virtual brain twins, what they are, how they are built, and the architecture behind them. Virtual means mathematical models in the digital world. Twins means it's personalized. It matches one specific person's brain. Virtual brain twins are personalized, generative, and adaptive brain models based on individuals' brain data. They use both anatomical data, like MRI, and functional data, like EEG. We start by estimate key parameters from the personal data. That's what makes the model personalized. Once we have the virtual brain twin, we can use it to make predictions. This predictive activity comes from a model's generative nature. It can generate new brain activity under different interventions. For example, in epilepsy, we can simulate different surgeries and predict which one might work best. In schizophrenia, we can test different medications and see how the brain might respond. And also, the model is adaptive. When the real brain changes after an intervention, the virtual brain twin can be updated so it continues to reflect the current state of that person's brain. Now let's talk about the models. In whole brain modeling, we typically work at two spatial resolutions. Low-resolution models use roughly hundreds of regions, depending on the atlas. For example, the VEP atlas has 162 regions, each covering about 10 square centimeters. High-resolution models are surface-based, reaching about 260,000 vertices at a 1 square millimeter resolution. First, let's look at the high-resolution modeling. We define the neural population activity at each vertices, denoted by psi, which depend on location x and time t. The first term describes the local dynamics. Each population interacts with its neighbors through local connectivity and with distant areas through global connectivity along white fiber tracks. And finally, we add a noise term to capture intrinsic variability. Now let's move to low-resolution whole brain modeling. At this scale, we aggregate neighboring neural populations into one single node. The local connectivity term no longer has meaning and is removed. From now on today, we will focus only on low-resolution examples. There are 5 main steps to building a virtual brain twin. First, we begin with anatomical data to construct the structural scaffold. 2, we place the neuronal models on each node. And 3, we map their activity to the sensor level using the forward solution. 4, sensor level data typically comes from functional recordings. So we then compared the model-generated signals with the real functional data to estimate the key parameters. Once those parameters are identified, the virtual brain twins is built, and 5 is ready for prediction. Now, I will walk you through each step. Step 1 is building the structural scaffold. Use T1 weighted MRI and a brain atlas. We defined the nodes, each representing one anatomical region. Then, use diffusion weighted MRI. We attract the network links based on the white fiber tracks. This gives us the first level of personalization, an individual structural scaffold. In the second step, we place the neural mass models on each node. This term is for their local dynamics, and this one is for the influence from the network. To illustrate this, let's take an example of epilepsy. We put epileptic equations on each node. Then we simulate the brain activities. When some regions have seizures, while others not. This level of activity is what we referred as source level. The next two steps build on two key concepts, source and sensor. This is our high-resolution source simulation on the right. Source activity represents what happens inside of the brain itself, independent of any measurement system. What we can actually measure and observe, however, are the signals recorded at the sensors. The sensors can be scalp EEG, stereo EEG, fMRI, or MEG. In step 3, we calculate the sensor signals from the source activity using the forward solution. In step 4, we inform the source activity from the measured sensor signals. To explain you how, here's an example using stereo EEG, where electrodes are implanted inside of the brain. We use CT scan together with T1 MRI to determine exactly mapping between SEEG sensors and then corresponding brain regions. Then we map the source activity using this equation to calculate the signals for SEEG sensors. Now, from a patient's SEEG seizure recordings, we attract the data features and estimate the key parameters using a machine learning algorithm so that the simulated activity best matches the empirical signals. In the context of epilepsy, now we identify the key regions capable of generating seizures, which we call epileptogenic network. We then formalize this result and highlight the regions on MRI, provide clinicians with information for diagnosis. The final step is prediction. If we are planning a resection and the target spots show up in blue, we can run a simulation to see if the patient might actually end up seizure-free. But in this case, the model says the seizures drop, but not completely stop. Let's recap the 5 steps. 1. Building the structural scaffold. 2. Assigning the neural mass models. 3. Computing the forward solution. 4. Inferring the key parameters. 5. Finally, making predictions. We will see an examples. of how virtual brain frameworks works for epilepsy. Under the same architecture, we can extend this approach to many other conditions. Here I show 6 examples. The key idea is to choose the control parameters that corresponding to the underlying pathological mechanisms. For epilepsy, the relevant control parameters is the regional excitability of the brain regions involved. For Alzheimer's diseases, the parameters can be related to regional beta amyloid and tau. For aging, we focus on global connectivity changes that we consistently observed in empirical data. For multiple sclerosis, a meaningful parameter is the time delay between regions due to changes in myelination. In Parkinson's, link weights act as control parameters because neurotransmitters problem in basal ganglia disrupt communication. For schizophrenia, the control parameters can be regional, such as gray matter volume changes, and also network level, including anomalies related to dopamine and serotonin systems. In terms of applications, epilepsy is currently the most advanced. The first clinical trials began in 2019. For schizophrenia, our first clinical trial is planned for 2026. In the upcoming tutorials, we will focus on epilepsy, brain stimulation, and schizophrenia. It's a huge team effort that brings us here today. Our team and our institute, led by Victor Yekza, and a special thank you to the TVB communications team to encourage me to create this series of tutorials. Thank you.